Volumetric analysis in the pharmaceutical industry. With an introduction to volumetric analysis in the pharmaceutical industry. Volumetric analysis is a fundamental technique in analytical chemistry that plays a vital role in the pharmaceutical industry. It is commonly used to determine the concentration of substances in pharmaceutical products, helping to ensure the safety, efficacy, and quality of medications. In hospitals and laboratories, it is also used in diagnostic tests to measure components in blood or urine, such as glucose or electrolytes. These accurate measurements are essential for proper drug formulation and dosage, which directly impact patient care and treatment outcomes. Now, let's move on to the theory. Volumetric analysis or titration involves a chemical reaction between a titrant of known concentration and an analyte of a known concentration. The purpose is to find the exact volume needed for the reaction to reach the equivalence point. In the pharmaceutical industry, this method is applied in acid-base titration to determine the concentration of active ingredients in medicines such as aspirin, antacids, or paracetamol. For example, titrating a paracetamol tablet with hydrochloric acid helps verify its neutralizing strength.
Yeehaw! This is why you need titration in your science toolkit. It's the key to solving chemical mysteries. Titration is a lab technique where a solution of known concentration is used to determine the concentration of an unknown solution. Imagine you're adding drops of a solution until a color change occurs. That's when you know you've hit the endpoint. Why is this important? Well, titration helps scientists in various fields, like chemistry, biology, and environmental science. They use it to analyze substances, create medications, and monitor pollution levels. Here's a quick rundown. You start with a solution of known concentration, called the titrant. Then you add it to the unknown solution slowly, watching for a reaction. The point where the reaction is complete is called the endpoint. From there, you can calculate the unknown concentration. Pretty cool, right? Next time you're in the lab, remember, titration is your go-to method for precise measurements. Stay curious and keep experimenting. Like and subscribe for more 1-minute education.
Okay, for the basic concept of calculation is you can see acid react with base, produce salts and water. And then for the formula you can use is MAVA, which is molarity of acid and volume of acid over 1000 over MBVB, molarity of base and volume of base over 1000 equal to A over B. Next, MAVA, which is molarity of acid times volume of acid over MBVB, molarity of base, times volume of base equal to A over B. And then if you ask to find mass, the formula you can use is mass equal to moles times molar mass. And for the pH calculation is pH equal to negative log, and then you can whatever value you can get, you can put at the H plus there. And then for the second formula you can use is pKW equal to pH plus pOH. Okay, for the calculation for back titration is for the first formula is mole reacted analyte equal to mole used analyte minus mole back titrated. Next, mole back titrated analyte equal to mole titrant times factor. And then
in direct titration, the titrant was added directly to the analyte until the reaction is complete. After that, the indicator was added inside the analyte. Direct titration used when the endpoint of titration can easily be obtained. Meanwhile, in back titration, the difference was a sample was added inside the analyte. After that, the indicator was added inside the analyte. Then the titrant was added directly to the analyte. In back titration, it used to determine the exact endpoint when there is a sharp color change. The purpose of back titration is the analyte may be in solid form. The analyte reacts slowly with titrant in direct or forward titration. The reaction with the intermediate reactant can be speeded up and reaction can be completely saved by heating.
Next, let's look at the applications of volumetric analysis in the pharmaceutical industry. First, the determination of vitamin C content using redox titration. Vitamin C in pharmaceutical syrups is analyzed using iodine titration, where ascorbic acid reduces iodine to iodine. This ensures the correct dosage in supplements. Second, the detection of metal ions in biological fluids using complex volumetric titration. Calcium or magnesium levels in blood or urine are measured using EDTA as the titrant, which helps diagnose conditions like kidney disease or osteoporosis. Third, drug purity analysis using back titration. This method is useful when direct titration isn't possible, especially for drugs that are poorly soluble. Back titration helps calculate the drug content based on the amount of excess reagents. Lastly, the determination of glucose levels in diabetic test kits. Some clinical methods involve titration reactions to measure glucose concentration in blood serum samples. So, those were just a few examples of how volumetric analysis is used in the pharmaceutical field, showing its importance in both drug manufacturing and clinical diagnostics.
Back titration is a type of acid-base titration. It is used to determine the concentration or amount of active substance in medication. As we know, some medications are in tablets. Hence, this titration technique can be conducted to determine the amount of active substance in a medicine sample. This is to ensure that the medications have correct dosage and is safe to consume. Paracetamol or acetaminophen has a phenol group that reacts slowly and incompletely with sodium hydroxide. So, instead of titrating it directly, we use back titration. Let me explain the methodology used to conduct a back titration to determine the amount of active substance, which is paracetamol, in a 500 mg paracetamol tablet. First, a sample of 500 mg paracetamol tablet was taken and crushed. Then, the sample was transferred into a 250 ml conical flask and a small amount of distilled water was added. Then, a known amount of excess hydrochloric acid was added into the conical flask to let the hydrochloric acid react with the basic sites on the paracetamol. The molarity of the HCl used was recorded for further calculation. Then, the conical flask was gently heated until all the paracetamol are completely dissolved, and the solution was allowed to cool to touch. Few drops of phenolphthalein were added as an indicator. Not all of the acid was neutralized by the paracetamol. Some remained unreacted. So, a titration process was conducted where the excess acid was titrated with a non-molarity of sodium hydroxide. This is to let the leftover acid reacted with the sodium hydroxide. In this process, the added acid is neutralized by the paracetamol tablet and the remaining acid is neutralized by the sodium hydroxide added. This is why it's called back titration. Then, the titration was stopped when the endpoint is reached, which was when a temporary faint color changes of the analyte obtained. Then, the volume of sodium hydroxide used was recorded for further calculation. Then, the procedure was repeated 2 to 3 times to find the average volume of sodium hydroxide used. Lastly, the calculation of back titration was calculated to obtain the last mass or concentration of the sample. This is how calculation of back titration can be done. In general, back titration will have two balanced chemical equations because of the two reactions that happen in the conical flask. After both of the equations determined, then you can step into the calculation. First step is calculate the number of initial moles of hydrochloric acid to 4 significant figures using the molarity and the volume of the hydrochloric acid solution. This is the total amount of acid requiring neutralization by both of the sodium hydroxide and the paracetamol tablet. Second step, calculate the numbers of moles of sodium hydroxide used to neutralize the leftover hydrochloric acid using the molarity and the average volume of the sodium hydroxide solution added. This is the numbers of moles of hydrochloric acid that was neutralized by the sodium hydroxide. Next is the third step, which is to determine the numbers of moles of hydrochloric acid that reacted with the paracetamol by subtracting the total mole of acid initially in the conical flask with the moles of acid that neutralized by the sodium hydroxide. Next, the fourth step is, from the equation 1, using the ratio of the equation, you have to determine the mole of paracetamol present in the reaction. The last step, which is the fifth step, is using the moles of paracetamol, find the mass of paracetamol by multiplying the molarity and the molar mass of the paracetamol. With this calculation, the amount of paracetamol present in the sample can be determined. The amount of paracetamol obtained indicates the dosage of paracetamol contained in a tablet sample. Pictures shown here are the list of correct dosage of paracetamol that is safe to consume by a particular person depending on their ages. This dosage list was officially published by the Pharmaceutical Service Program, Ministry of Health Malaysia on their official website. So, if the mass or amount of paracetamol determined in a sample of 500 mg paracetamol tablet is within the claimed dosage on the label, then it is safe to consume.
Hence, to conclude is, volumetric analysis is a chemical analytical procedure based on measurement of volume of reaction in solution. It uses titration to determine the concentration of solution by carefully measuring the volume of one solution needed to react with another. In this slide, we can learn about the theory, methodology, and calculation of volumetric analysis. Hence, volumetric analysis plays a vital role in the medical field by enabling precise measurement of chemical substance in body fluids and pharmaceutical products. This technique is essential for determining the concentration of drugs, diagnosing diseases, and ensuring accurate formulation of medication. Through titration methods, medical professionals can monitor patient health by adjust dosage appropriately and maintain safety and effectiveness in treatment.
If
If there are any questions, please comment below in the comment section. Thank you for watching. Bye.